Namaste friends, welcome you all for the biology session. Let us continue with the chapter cell cycle and cell division. In the last part we have completed mitosis and we have some remaining questions. Let us solve some questions related to mitosis and we can take meiosis. So during anaphasic movement, chromosomes dash of each chromosome is or are towards the pole and dash of chromosome trail behind, means they're going to left behind. So if you consider this generally, this is a particular pole, two poles of a cell, and here the chromatid will be there, and where the chromosomes which will be there, they are the in which sister chromatids will going to separate. So this is how the spindle fibers will be attached with the centromere. So centromere is towards the pole, right, whereas arms are towards the equator. So answer for that we can say that centromere of each chromosome is or are towards the pole and arms of chromosome trail behind, means that will going to present behind this centromere. So this is the explanation for this, and answer is centromere and arms. A is the answer. Next, select the correct match. So quiescent phase, as we know that is generally cell arrest phase, that is G0 phase. So this is the wrong answer. And synthesis phase, that is S phase, where DNA duplication occurs. The DNA duplication, the chromosome number remains same but chromosome content increases. Centromere splitting, so here we can say that the centromere splitting in anaphase, just now we discussed that where the chromosomes will going to split. Here we can say that the chromosomes will going to split and forms a two sister chromatid. This is one sister and second sister chromatid. And whereas here in this case, before that the centromere divides longitudinally. So one centromere will be there in the chromosome and that centromere will going to split longitudinally and that forms the two centromere, and centromere splits in anaphase. So next option, chromosomal condensation telophase. Here the condensation takes place in the metaphase and initiation in the prophase. But whereas in telophase we can say decondensation occurs. So this is also wrong. So answer for that is C. And 23rd question, match 1 and 2, column 1 and 2, and select the option from the given codes. So here just now we discussed that while during anaphase, chromosomes in which they are going to split and forms a chromatid, and here sometime it will be V shape. So in this case, here we can say in some time V shape, and sometime, that is, as we all know that there are four types of the chromosomes, that is metacentric. So that is what the V shape, and sometime the submetacentric. So generally one side which is a little longer, that is L shape which will give, and this is generally V shape, right, metacentric V shape. And whereas here L shape, so one is longer and other one is little longer, and here we can see like this, which will be so like this, so which will be going to move towards the opposite fold sometime. So it will be in different cells, and submetacentric, metacentric, submetacentric, and acrocentric. So acrocentric means, that is, here we can say that is a J shape, right, generally that is J shape, right. So which is towards, here the L one is long and J shape is one is completely long, the other one is very the short, completely short. So here this is a L shape. So here we can say L shape, and in some it is J shape, right. So one is completely long, other one is slightly very short, and in some it is I-shaped. So that is where we can say that the centromere towards the pole and chromatids which will be, so this is during what is it, the anaphase. So V-shaped exactly V-shaped we can say metacentric, submetacentric L shape, and acrocentric J shape, and the last one telocentric, so telocentric which is I shape. So answer for that is V shape metacentric, that is A2. So from these two options we can select. And L shape, so L shape, that is submetacentric, B4, right? So in both we have B4. And J shape, that is acrocentric, D3. So here we can say that the C1 and D3, I shape telocentric. So answer for that is B. So this question we can expect VLJI. Just you remember that V that is metacentric. In order we can say metacentric, submetacentric, acrocentric, and telocentric. Based on the position of centromere, we can classify the chromosomes into 4 types. So answer for that is B. Next question. So 23rd question, answer is B. Next, 24th question. Select the incorrect match regarding mitotic cell division. So prophase, the chromosome begins to uncoil. So here we can say that it won't, it begins to uncoil, that is it begins to coil, right? Or you can say that the coiling takes place. That is what it, initiation of condensation, packaging of the chromatin fibers, right? Metaphase chromatids move apart. So metaphase chromatids move apart. No, chromatids will going to align at the equatorial plate. So this is also a wrong statement. And telophase, the nuclear membrane reappears. This is correct. And next, late anaphase, each chromosome consists of 2 chromatids. So each chromosome consists of 2 chromatids. So in the late anaphase, each chromosome consists of, generally we can say that the 1 chromatid. So that is in the anaphase. So wrong. Interphase, the chromosomes are not distinct. Yes, that is also correct. So here we have to select the incorrect statement. So what are those incorrect statements for this generally? So second one is incorrect and fourth is incorrect. So second and fourth statements are incorrect. So answer for that is B. Answer for that, here we can say, first one, this is incorrect. So already we mentioned that is that chromosomes begin to the coil, that is condensation. So this is incorrect. And metaphase, they won't move apart, our alignment will be there. So and the second one, so second one is also incorrect, right? And the next one, prophase, which is uncoil, begin to the uncoil. And metaphase, chromatids move apart, that is a wrong one. And this is right one. And fourth one, which is the wrong one. So here we can say that the first, second and third are the wrong statement. So first, second and fourth. So here we can take one more, this one. So first, second. So in prophase, chromosomes begin to uncoil. So all these are, in prophase it begins to coil. When it begins to begins to uncoil, we can say that is in telophase. It is in telophase. So one more option here we have to take 1, 2 and 4 or wrong statement. Right. Next, what is true about telophase stage in mitosis? What is true about telophase stage in mitosis? So what happens in this case? Chromosomes lose their identity as a discrete elements. That is what, in which the anaphase, they move completely towards the pole, right? So here which will going to, the decondensation, condensation, which will going to uncoiling, takes place. And nuclear, the envelope reappears, all those materials, the organelles will going to reappear. So decondensation. So condensation, decondensation. So in this, at the end here we can say that the one cell, each daughter cell will be with one centriole, and those Golgi complex and then nucleolus, nuclear membrane, endoplasmic reticulum will going to reappear. So and chromosomes will going to cluster at the opposite pole. So this is the characteristic feature of telophase. Any chromosomes clusters at opposite spindle folds. That is correct. And then nuclear envelope, nucleolus, Golgi complex, endoplasmic reticulum reforms. So this is a characteristic feature of telophase. All of these are correct. True about telophase. Next, direction. Refer the given figure and answer 26th to 29th question. So let us take the answer for this. So here we can say that by seeing this, we can say easily that is a C is prophase, where condensation will going to take place, and this is a metaphase, B is a metaphase, right, and A is anaphase, and D is the telophase together with cytokinesis. So simultaneously cytokinesis occurs. So let us take, identify the given stages of mitosis and select one. So B is prophase, A is anaphase. So here we can say that A is anaphase and B is metaphase, and C is the prophase, and D is telophase. So answer for that is C. So C is the correct answer for that 26th one. So next, anaphase, metaphase, prophase and telophase. Next, which of the following shows a correct sequence of the given mitotic stage? So mitotic stage, here we can say, we can align here only. So the first prophase, right? So prophase, next CB metaphase, and then anaphase. CBAD is the proper alignment, or you can say that this sequence CB, AD, CBAD. So answer for that is D is the answer. So CBAD, where exactly the alignment takes place. And next, at which of the given stage of mitosis chromosomes appear in VLJI shape? So generally we know that VLJI shape, V is metacentric and L is submetacentric, J is the acrocentric, and I is telocentric. So here you can say that the metacentric and then submetacentric, and J is acrocentric and I is telocentric. So this is exactly during the anaphase. So where the chromosomes which are going to divide, right? So chromosomes divide and the single chromatid starts moving towards the opposite pole. So this will be seen in anaphase. So anaphase is represented from in the diagram, that is A. So answer for this is the A is the answer. A phase which represents the anaphase, right? Next, read the given statement which represents the feature of figure ABCD, match them correctly and select the correct option. So by seeing that the figure ABCD which was given, let us take with respect to the diagram. The chromosomes appear like the ball of wool, right? You can say that this pyreme stage. So chromosome appear like ball of wool. So generally in which we can say that the chromosome appears like a ball of wool. So if this is one, it appears like a ball of wool which will be there in the prophase. So here we can say that in the diagram we can select which is a prophase, that is C is the prophase. So answer for that, in the given figures A, B, C, D, match correctly, match and select. Chromosomes appears like, so that is a fourth stage. We'll take A4. And reformation of nuclear envelope, nucleolus, Golgi complex, so which will be takes place. So that reformation which will be takes place in, we have to arrange the changes which are going to take place. So first one, what is that? splitting of centromere in anaphase. Let me write here splitting of centromere in anaphase and formation of equatorial plate, that is in the metaphase, and reappears takes place in the telophase, and this which appears in the prophase. So here we can say that arrange that prophase, splitting of centromere. So read the statement and statements in which represent the future of figure ABCD, right, and match them correctly and select the correct option. So ABCD diagram. Let's take options here. Splitting of centromere in anaphase. So C is prophase, B is anaphase, D is telophase, anaphase, metaphase, prophase, metaphase, and anaphase, and CB, DA is telophase. Now we can say that is prophase A4. So this is splitting of centromere which takes place in the anaphase. So D formation of equatorial plate, metaphase. So answer for that here we can say that is exactly which takes place in A4. So this is centromere splitting which takes place in anaphase, right, and B3. So B represents telophase, B represents, that is the equatorial plate, that is metaphase, right. So let me write here, just A means diagram, right, and this is this characteristic feature A, sorry for that. So A we can say that diagram, right. So A diagram represents anaphase, and B represents metaphase, C represents generally the prophase, right, and D represents C1 which represents the prophase, and D represents telophase. So telophase. So answer for that is the A, right. Next. Let us take next option. So which phase of mitosis is essentially the reverse of prophase in terms of nuclear changes? So reverse of prophase means what is that? Generally condensation starts where condensation starts in the prophase, right, and this is the one. All those the organelles will going to disappear. So all these things will going to reappear in the telophase, and along with that where condensation occurs, that will going to give once again uncoiling takes place. So that is in case of telophase. So answer for that is C. And cytokinesis in an animal cell takes place by a method of, so generally we have discussed this, that is what is exactly the cleavage is and where the cleavage furrow takes place. So cytokinesis animal cell is because of the cleavage furrow. So this is what we can say, this is a cleavage furrow where the plasma membrane which will be pulled by centriole and that will going to attach, and that forms a complete division, that leads to complete division of this cell. So when this furrowing takes place, that is during cleavage, this is a furrowing. So furrowing, right, so direction, furrowing method in centrifugal, that is towards the center. So this is furrowing centrifugal, furrowing method, that is centripetal direction, while the plant cell, so here you can say that this is towards the center, that is centripetal. Furrowing, that is towards the center, that is centripetal. And in case of plant cell where there is no generally there will be a cell membrane, plasma membrane will be there, the cell wall which is very rigid, so it can't form a furrow, so that the Golgi vesicles will going to form a phragmoplast, and that will going to extend it outward, so towards the periphery, that is called as centrifugal. So this is the formation of cell plate. So these vesicles are called as phragmoplast, and those phragmoplast later develops into cell plate, and that cell plate which will going to move towards the periphery, that is cell plate and centrifugal. So answer for that is, so here we can say that the 31 question, so answer for that is furrowing centrifugal, cell plate, centrifugal. So this cell plate and further develops into, what is it, this cell wall. So cell wall formation in case of, what is it, the plants. So that is, we can say that the cell plate formation, with the help of cell plate formation. Here it is a cleavage furrow, so answer is B. Right, and spindles usually persist in the form of, so generally these here the spindles directly attached from the center towards the plasma membrane, and they're going to pull, constrict, and here it will move towards the inward, and here it will move towards the outward. So here we can say those vesicles which will going to form. So with the help of that spindles, right, they're going to, what is it, the form arrangement in the form of plate. So this is where the cell plate formation takes place. That is what we can say that phragmoplast. So spindle usually persists in the form of phragmoplast. So what are those phragmoplast? If they're going to ask the answer for that, the phragmoplast, these are, what is it, the forms during cytokinesis. In cytokinesis, formed during, this is a form of phragmoplast, right? Phragmoplast will take, what exactly the phragmoplast during, what is it, the cell plate formation. So during, what is it, the cell plate formation, phragmoplast during cell plate formation, what happens? So here the phragmoplast forms during cytokinesis, that is correct, and these are the assembly of microtubules. This is a includes assembly of microtubules. Along with that, what is it, the actin filaments. Along with that actin filaments, that is what we can say that which is going to form actin filaments, is going to form a spindle. So spindles, which includes, what is it, the generally the assembly of microtubule, actin filament, and some molecules which acts as a, what is it, the framework, which acts as a, what is it, the framework. So along with some molecules, right, which acts as a framework for cell plate, formation. And this cell plate, which is going to, what is it, the assembled, and later that will be formed, leads to the formation of cell wall. So this structure which will be there generally, which will going to form, that is what the phragmoplast. So phragmoplasts are, which are the assembly of microtubules, actin filaments, and molecules. So altogether the phragmoplast that forms a framework for cell plate and finally forms into cell wall formation. So answer for that is B. 32, answer for is B. So meiosis. So before going to take meiosis, let us take what exactly the meiosis. As we all know that meiosis is, that is the reductional division, which takes place in sex cells or, what is it, the gamete mother cells. Reductional division which takes place in, what is it, the sex cells or gamete mother cells, which leads to the formation of, which helps in the formation of haploid gametes. In almost all organisms, the haploid gametes which are going to produce through the process of, what is it, the meiosis. So let us take this, the meiosis, right, which includes, where we can define like this, it is a division of diploid germ cells, that is what we can say sex cells, right, which leads to the formation of haploid gametes. So here this occurs during gametogenesis. So what exactly the meiosis includes? It includes meiosis 1, whereas in mitosis, only mitosis, right, it includes the prophase, metaphase, anaphase, telophase. Here meiosis 1 and meiosis 2. So meiosis 1, which includes prophase 1, metaphase, anaphase, metaphase 1, anaphase 1, telophase 1. And later here the meiosis 2, which includes prophase 2, metaphase 2, anaphase 2, and telophase 2. So this meiosis exactly resembles the, what is it, the mitosis. Exactly resembles the mitosis, right, which takes place in the somatic cell. Whereas here, some changes will going to take place. So in meiosis 1, mainly the prophase 1 is the largest phase. Not, you can say largest phase, that is the longest phase. Better to say that is the longest which is the longest phase. So why it is called as longest phase? So prophase 1, which includes 5 subphases. So prophase 1, this is very important, we can expect a question here, right? So prophase 1, so which includes 5 subphases. So among those 5 subphases, so if you're going to arrange that in order, right, so A, which will be called as leptotene, and B is zygotene, pachytene, diplotene, and diakinesis. So leptotene, zygotene, pachytene, diplotene, diakinesis. So what are the characteristic feature here? So here we can take the unique feature of all these subphases of, the subphases of prophase 1. So here this phase, which will be considered as thin thread. So why condensation starts here? So here the thin thread like appearance will be there. So here the condensation starts. So where we can say that the chromosome begins to condense. So this is very important. Definitely we can expect or arrange that phases in series. What are the subphases of prophase 1, or what is the uniqueness of that particular phase? Leptotene, chromosome condensation begins. And zygotene, where the condensation starts, and here the pairing of the chromosomes occurs. Hence it is called as paired thread. So in this we can say that the paired thread, where the chromosomes closely paired. So closely paired. So pairing of chromosomes occurs, that too which are exactly the similar. So that similar chromosomes, we call it as homologous chromosomes. So pairing occurs with the help of a complex called as synaptonemal complex. Synaptonemal complex. So zygotene is a paired thread, closely paired, and synaptonemal complex, right? Because why the pairing takes place in the the zygotene phase, that is because of synaptonemal complex. So that is what you have to keep it in mind. Synaptonemal complex, which helps in the formation of homologous chromosomes. So here we can take the diagram for that, right? So that is zygotene, where you can say that the pairing of homologous chromosomes occurs. So these are the chromosomes, sister chromosomes, and here the sister chromosomes, right? Sorry, exactly, that is equal. So we'll take that equal length. So so if you consider that here with the help of synaptonemal complex. So this is like the zipping action where it will going to takes place. This is a synaptonemal complex which forms, and that will going to the join those. So that is what homologous chromosomes. In zygotene, formation of homologous or fusion of synapses occurs, right? So pairing of similar, that is homologous chromosomes with the help of synaptonemal complex. With the help of synaptonemal complex. So that is what. So that condition where it will be formed, right, where it will give two the chromosomes, that is bivalence, right? So bivalence, what is it? Homologous chromosomes. And here the tetrads are, you can say bivalence means paired homologous chromosomes. Bivalent means paired homologous chromosomes. Where exactly they are going to appear? That is in the zygote stage. But here in these, the chromatids are there but not clearly visible. Not the clearly visible. Formation occurs but not clearly visible. So let us take next phase, that is a packaging phase. So packaging, where complete synapses occurs. Here in the synapses starts in zygote, synopsis, the initiation of synapses, right? So pairing of homologous chromosomes. Here synapses completes, or completes completely, right? So synapses completes, and where you can say that bivalent splits. So bivalent splits. When bivalent splits means chromosome splits. Here we can show like this diagram. For this we can show like this. So this will going to split like this. Bivalent splits. So there will be the attachment, right? And here chromatids can be clearly visible. See, in this diagram you can say they are attached here in this portion. So here the we can say that the one chromatid, second chromatid, third chromatid, fourth. You can say that 1 and 2 in each chromosome. So total 4. So here that is why we can say tetrads are clearly visible in the packaging stage. So why that packaging stage tetrads are clearly visible? So here you can say that the bivalent splits, right? And this, which generally forms it leads to the formation of similar chromatids, right? And these chromatids will going to consider as a tetrads, as there are 4 and 2 in each chromosome, 1 and 2 of this chromosome, 1 and 2 of the second chromosome. These are homologous chromosome. So that is why you can say clearly visible. And next, here we can take one more change where the crossing over takes place. So just to one point we can change here. So as the synaptonemal complex they starts disintegrate, so where the chromatids, that is a non-sister chromatids, will going to the fuse, or you can say that the crossing over starts here. So in packaging, crossing over starts. So crossing over starts, right, with the help of, why exactly the crossing over occurs here? At the point where crossing over occurs, there is an enzyme called as recombinase enzyme. So the point where the recombination occurs, so assume that this is a point, here is this point at which the crossing over takes place, that is called as recombination nodule. Recombination nodule, right? So that is at which the point at which crossing over occurs. Point at which crossing over occurs. So this is a recombination nodule. Where exactly? Why exactly at that point? Because of the presence of enzyme which helps in the recombination called as recombinase. Point to be noted. Enzyme which involved in the crossing over or recombination. Recombination because of the crossing over, right? So point recombination nodule is a location where the crossing over occurs. The enzyme which is present, that enzyme which is called as recombinase enzyme. So that is what you have to keep it in mind. So what is that? The crossing over, exchange of the genetic material between non-sister chromatids of 2 homologous chromosomes. What is that crossing over? Crossing over means, that is in short form I will write, exchange of the genetic material, exchange of genetic material between 2 non-sister, or I can say that the non-sister chromatids, chromatids of 2 homologous chromosomes, or homologous chromosomes. We can say like that, 2, or we can say that is 2 homologous chromosomes. So that is what crossing over. So crossing over with the help of enzyme called as recombinase, where the point where exactly that recombination occurs, that is called as recombination nodule. ಇಲ್ಲಿ ಕ್ವಶನ್ ಕೇಳ್ಬೋದು, ಯಾಕೆ ರೀಕಾಂಬಿನೇಷನ್ ನಾಡ್ಯೂಲಲ್ಲೇ ರೀಕಾಂಬಿನೇಷನ್ ಆಗುತ್ತೆ? ಬಿಕಾಸ್ ಆಫ್ ದಿ ಪ್ರೆಸೆನ್ಸ್ ಆಫ್ ಎನ್ಸೈಮ್ ಅಟ್ ದಟ್ ಪರ್ಟಿಕ್ಯುಲರ್ ಲೊಕೇಶನ್. ಆ್ಯಂಡ್ ನೆಕ್ಸ್ಟ್ ಇಸ್ ದಟ್ ದಿ diplotin. So diplotin, here we mentioned that in the packetin, that is a thick thread where complete condensation will be going to take place. Here I will mention that is thick thread. So thick threads. So here pairing in zygote and thin threads, slight condensation, slightly initiation of condensation here, right? Pairing, synapses occurs, paired thread, and here is a thick thread, right? Because of what is it, the crossing over, that is thick threads. So thick threads. And diplotin, here we can mention that in diplotin generally 2 threads will be clearly visible, right? And synaptonymal complex in this condition diplotin, right? So it will going to start dissolve, right? And in diplotin, right, where exactly the next phase, that is D, in diplotin synaptonymal complex, what is that, the dissolution occurs. So here we can say that where exactly the point of crossing over takes place, and that leads to the formation of X shaped structure that is clearly visible in the diplotin stage. So if this is the thing, this is called as chiasma, or you can say that chiasmata. So chiasmata, or you can say chiasma, is visible, clearly visible. So clear appearance of what is that, chiasma or chiasmata. So clearly visible, and here you can say that the dissolution starts. So initiation here and the partial dissolution of, there is a maximum dissolution of synaptonymal complex occurs. So and finally we can say that the where here chiasma clearly visible and dissolution starts. And in the last, what is it, the phase where complete dissolutions and the terminal, it will going to move towards the terminal here, from here to what is it, at the point. So here X shape. So chiasma, what is it, which is visible here in the diplotin, but it chiasma, which what is it, the formation occurs in the packetin stage. So chiasma appears, keep it in mind, chiasma appears in packetin stage but clearly visible in, what is that, clear appearance in diplotin, and that chiasma disappears. So chiasma terminalization. So terminalization of chiasma, that is in diakinesis. So keep it in mind. So diakinesis, terminalization of the next phase of the, so here terminalization, complete edge move agutte, that is terminalization of chiasma. So what exactly happens there, terminalization of chiasmata, right, where they're going to, what is it, the exchange, after exchange they're going to free, but sometimes they won't be, right, complete here, no synaptonymal complex. You can see that no synaptonymal complex, where at one particular point where it forms a X-shaped structure. Actually chiasma is X-shaped structure which appears in packetin. So chiasma, that is X-shaped structure but clearly visible in the diplotin, and terminalization of chiasmata takes place diakinesis. Adike simply to remember that we can say diakinesis, right? Dying of chiasma anta simple ಆಗಿ ಹೇಳ್ತಿರ್ತೀವಿ, diakinesis ನೆನ್ಪಿಟ್ಕೋಬಹುದು. So chiasma is actually X-shaped structure, right? So in the last stage you can say that the diplotin stage, where in this, what is it, the last stage, that is diakinesis, where mitotic spindle fibers originate. So if this is a cell, and here which will going to takes place a mitotic spindle fibers, or from this pole also we can write, no problem. So spindle fibers arranged. So when mitotic spindle fiber starts, so bivalent is ready. So these are called as bivalents, and these bivalents are ready to enter the metaphase. So from diakinesis it enters the metaphase 1. So what happens that generally we know that what exactly the metaphase, that is there we used to mention the chromosome alignment in the metaphasic plate, but here sometime there will be attachment with, what is it, the terminal end, or you can say telomere, or sometime it may not. So alignment of homologous chromosomes, right? Alignment of homologous chromosomes in the metaphase 1. So unique feature of that metaphase 1, bivalence at equatorial plate. Not, we won't say that the single chromosome, we'll say homologous chromosome. As we all know that paired homologous chromosomes are called as bivalent. So bivalence, what is it? At equatorial plate. So that is equatorial plate. That is, what is that? We can say characteristic feature of metaphase 1. So spindle fibers attached to the centromere of each chromosome. Relatively, you know, chromatid. So now in the anaphase 1, what happens if you consider the next phase, where the chromosomes, what is it, this with respect to, what is it, the centromere, they are going to move with their both chromatids. So just recollect that in the anaphase, just we used to write like this, chromatids will going to move towards the opposite pole. But here you can see that is the homologous chromosomes will going to move towards the opposite pole. So means only a chromosome, single chromosome is going to move. And telophase, what happens? Once again that cleavage for row, right? And this will going to, what is it, decondensed. So here two, what is it, the non-sister chromosomes are there. Two non-sister chromosomes. That is what which leads to variation. So why that variation? Because of crossing over during packaging stage. So here we can expect this question, in which phase, what is it, the crossing over occurs? That is in the packaging stage. And what is it, synaptonemal complex seen in, what is it, the which helps in synapses, that is in, what is it, zygote. And crossing over takes place in packaging, and chiasmata clearly visible, right? Clearly visible generally during, what is it, the diplotin stage. So packaging chiasmata appears, and in diplotin the chiasmata clearly visible, and whereas in diakinesis the complete, what is it, the terminalization of chiasma occurs. Sometime it may be attached. So that is here at the end of this, that is in the diakinesis, the nuclear envelope, exactly the prophase end, late prophase ಅಂತ ಹೇಳ್ತೀವಿ, nuclear envelope, what is it, the Golgi apparatus, Golgi complex, and then endoplasmic reticulum disappears, right? So disappears, same thing, right? This is prophase. And next, after that prophase, it enters, what is it, the after the diakinesis, it enters the metaphase 1, and then metaphase 1 it enters the anaphase 1, anaphase 2, telophase 1. So here with the, what is it, the non-sister chromosomes. Now this phase will going to enter, that is prophase 2, which is exactly like, what is it, the similar to the, what is that, prophase of mitosis, similar to the prophase of mitosis. So there you can recollect, I have already explained. So next, so let us take some questions here. So and then prophase 2, we know that chromosome, again condensation starts here. So two cells which will be there, and complete cells it will going to resume before going to divide, and these cells will going to enter the, so what is that, here decondensation, here condensation initiates. So prophase 2, and then metaphase, alignment of chromosome, not homologous chromosome, only chromosome, right? So that is, we can say non-sister chromosome, which are in this condition. So if this is a cell and this is a B cell, let me take only A cell. So decondensation initiates, sorry, condensation initiates, right? So that is coiling. And here, what is it, the alignment of chromosome, that is metaphase 2, exactly like metaphase of mitosis. And here from these chromosomes, chromatids will going to split. So that is what difference between anaphase 1 and anaphase 2, metaphase 1 and metaphase 2, exact the segregation of, what is it, the genetic material or separation of, what is it, the chromosomes, and that represents, and finally this telophase, here non-sister chromatids. So that is once again decondensed. So that is telophase 2, telophase 2. 2. So by this we can complete the meiosis 1. So telophase 2, what happens? Two groups of chromosomes, what is it, will going to reappear, envelope will going to reappear, and finally you can say that this is the one cell will going to produce a 2 cell, and remaining B cell will going to produce another 2 cells. So haploid, you have only 2 chromatids, that means which represent one chromosome, not a paired condition, one chromosome unpaired. So idu one cell, idu one cell gadre hage B cell alli same. So this is a haploid condition. At the end 4 haploid cells can be seen. So this is what we have to keep it in mind during meiosis. So let us take some questions which are related to meiosis, right? And we can solve those questions. So let's take one by one. So let us see first question here for that, meiosis. So meiosis consists of 2 cell division without any DNA. Interphase is common only once, what is it, the cell will going to divide. Interphase is common, right? Here also we can say that is generally the cell which will going to divide and that will going to form. So what happens, 2 times cell division will be there. So 2 cell divisions in which chromosome number reduced to half. 2 cell division without any DNA replication, only once. 2 cell division once, or you can say 1 replication will be there. 1 replication will be there. So 2 cell divisions in which chromosome number reduced to half. 2 cell division with only 2 rounds of chromosome replication, only 1. Single cell division, no. 2 cell division with the chromosome replication. So answer for that is B. And next best material to study the meiosis. So as we all know that generally whenever we are going to select the best material where the complete division will be there. So in male and female gametes we can select that, or you can say male and female structures, reproductive structures. So in which where exactly we can say that the anther, microsporangium in plants, right? So anther, right? So in ovary where we can say that the ovum development takes place, but it is not a best material, only once, only one, what is the egg cell will going to produce, whereas here many, the microspores in anther, the microspores will going to produce. So that is what anther, which is also called as microsporangium, where microsporogenesis takes place. Microsporogenesis takes place. So microsporogenesis rapidly which takes place, sporogenesis which leads to the formation of what is it, the microspore tetrad. Microspore tetrad. So microspore tetrad will be with 4 microspores and finally forms of 4 pollen grains. So that is why best, what is it, the material to study is young anther. And the term meiosis was given by Farmer and Moore. So here one additional information, the term meiosis was given by Farmer and Moore, right? So Farmer and Moore who coined term meiosis. Coined term meiosis. So that is the one to keep it in answer, keep it in mind. So at which of the following stages the chromosomes appears single, thin thread-like. So I already mentioned that the chromosome appears thin thread-like structure, that is thin threads leptotin. So thin threads leptotin. So this is, you have to keep it in mind. So where exactly we're going to get the question from this. So leptotin is the first phase. LAZP dip dia. Leopard and zebra, right, playing in water, right, playing in water. Unfortunately zebra, right, can't be able to swim properly, so that is dip, right, sink in water and die. So LAZP, you can remember that LAZP dip dia, LAZP dip dia. So arrangement is in sequence. So that is what we can say: leptotin thin thread, zygotine paired thread, leptotin thin, what is it, the thread, zygotine paired thread, packetine thick threads, diplotin 2 threads, And finally we can say that diakinesis moving through, where terminalization of chiasma occurs. Terminalization of chiasma occurs. So let us take in 36th question. Zygote of prophase 1 is characterized by, just now we discussed, that is chromomeres, right? So chromomeres, that is end of the chromosome, that is a chromomere, wrong, right? So in zygote we can say synaptonymal complex. So zygote, what happens, that is pairing takes place. The process of pairing is called as synapsis. So with the help of synaptonymal complex. So crossing over takes place where exactly, that is packeting. Terminalization of chiasma, that is takes place in diakinesis. So 36. And crossing over in diploid organisms responsible for, just now I mentioned that the exchange of genetic material which leads to variation, that is what the basic, what is that, the process which leads to variation is sexual vital process, what is that, which leads to a variation, that is sexual reproduction, where you can say during gamete formation this leads to, what is it, the where crossing over takes place, that leads to the formation of recombinants. So with what is it, genetic variation. So responsible for, what is it, so in this generally we can say that it leads to recombination. So recombination of alleles. So if you consider, what is it, what are those alleles, right? So at particular loci, right, particular genes are present which is represented forms of genes, alternative forms of genes, that is these are alleles. So alleles, the place where they are located, the location is called as locus. Many loci. So recombination of alleles, that is what, the exchange of genetic material. So genes are represented in the form of alleles. So answer for that is D. And members of a homologous pair of chromosomes. So what are those, the members of homologous pair? Generally we say they are identical, right, in size and appearance exactly same. If you're going to write like this, you have to write, right, so we're exactly so let us take, means same size and same appearance. Here we can say same size and same appearance, exactly, which looks, or is that the similar in size and similar in appearance. So identical in size and appearance. So 38 A is the correct answer. So which of the following is a correct about bivalent? So as we all know that, what is the meaning of bivalent? Bivalent in which, what is it, the tetrads, so 4 chromatids will be present. So bivalence appears during, what is it, the synapses, or you can say that bivalence appears in zygote phase but clearly visible. The tetrads of the bivalents, what is the tetrads of the bivalents, clearly visible, tetrads of bivalents, so clearly visible during, what is it, packeting, during packeting. So which bivalents are tetrads? We can say that is the correct answer about, what is it, this one. So select correct about bivalents ಅಂತ ಕೇಳ್ತಿದ್ದಾರೆ. So what is that, the bivalents? We know that there are 2, what is it, chromosomes which are homologous chromosomes, so that is the first one which is correct, right? And then a bivalent means 4 chromatids, 2 centromere, that is correct. So 4 chromatids, this is a 1 and this is a second chromatid. So 1 centromere. In another chromosome, 2 centromere, 4 chromatids. So 4 chromatids, 2 centromere, that is also correct. One bivalent consists of 2 homologous chromosomes. So one bivalent which has 2 homologous chromosomes, that is also correct. And bivalence forms in zygote stage, so that is also, what is the correct. So 1, 2, 3, 4 are, what is the correct answer, what is that, the correct answer related to bivalent. So all these are correct answer, 1, 2, 3, 4. So let us take 40th question. So in this here we can say that is the process of crossing over is assisted by which of the following enzyme? I already mentioned that where exactly, what is it, the process of crossing over. Crossing over takes place during packaging, and that packaging, what is the crossing over exactly takes place, that is at the site which is called as recombination nodule, where there will be two enzymes generally, that is we can say that the polymerase and the endonuclease. So the process of crossing over is assisted by which of the following enzymes? So polymerase, that is a DNA polymerase, this is DNA replication. So here endonuclease, we are going to the cut particular segment. So if these are the two segments, and endonuclease will going to cut that segment, this is a B segment, and here will be the C segment. So C segment, and this will be removed. C segment which will be removed. So that will be cut, and here this part will be taken out, and so instead of this part they are going to take this to here. So they are going to add this segment to here. So that is what C. With the help of this they are going to ligate. So to cut they are going to use restriction endonuclease enzymes, that is what endonucleases, and to ligate, to join, that is they are going to use ligases. So answer for that is A and C. So both A and C are the correct answer. So next, to build next question. So here, to build up food reserves in the cytoplasm, the chromosomes become unfolded and starts transcription of mRNA and rRNA. So means where the protein synthesis will going to takes place through the process of transcription and translation. So when the process of transcription and translation takes place, right, the process of transcription and translation takes place, transcription, where we can say that DNA which will be converted into mRNA, and mRNA which will undergo translation to produce a protein or enzymes. So what is that? To build up food reserves in cytoplasm, chromosomes become unfolded and to start transcription of mRNA and rRNA during which phase of meiosis 1. So exactly at which phase this the process takes place, right? So at the end where it starts, the meiosis 1, the end in the meiosis 1, the prophase where exactly it starts recombination, and it will going to produce the variants, and that variated, or you can say that the recombinant chromosomes will going to show transcription and translation. So that is why generally in case of the diplotin, where terminalization takes place and ending, and during which phase of meiosis 1, you can say after diplotin starts can ready to undergo transcription and translation. So answer for that 41 question is diplotin. Right, at which, so next question. So diplotin stage of prophase 1 is characterized by, so diplotin stage of prophase 1 is diplotin means it is a fourth phase where dissolution of synaptonymal complex occurs, that is correct. Separation of synapsed homologous chromosomes except at the site of crossover, that is what X-shaped structure. The formation of X-shaped structure is called as chiasmata. So all of these will be there. That is what chiasma appears in packaging but clearly visible in diplotin stage. So all of these are correct answer. So to solve next questions, further questions, refer the following statement describing prophase 1 of meiosis. So if we're going to use a prophase 1, that is exactly which comes under meiosis 1. So answer the question 43 and 44. So thin thread like the chromosomes with beaded appearance. So that is where we can say generally which takes place in the leptotin stage. So this is where the beaded like appearance, that is in the leptotin stage, like this, which will be appears, right? So appearance of recombination nodule, as we all know that always the recombination nodule where the crossing over takes place, that is in the packaging stage. So leptotin we can say 1, packaging we can say 2, right? Formation of bivalents or tetrads. So bivalents are tetrads which will going to take place. So appearance of recombination nodule during packetine. So before leptotene, after that, so formation of bivalents, that is zygotine. So zygotine is second phase and this is third. Packetine is third. Termination, appearance of chiasma. Appearance of chiasma, what is it? The fourth phase, we can say that is generally which will be appear as terminalization of chiasma, that is diakinesis. Diakinesis and appearance in diplotin. So here if you're going to arrange that, zygotine, leptotene is 1, zygotine, that is, what is it, let me arrange this, appearance of recombination nodule, packetine, that is 2, that is 2, and formation of bivalent tetrad. So here this 1 become leptotene, so that is 1 which is leptotene on in all these options we have, and 2 is, that is the packetine recombination nodule. So packetine, so in these two we have to select answer. So leptotene, packetine 2, and third one formation of bivalents, that is in zygotine. So here we can say this is a zygotine we have, and terminalization of chiasma, diakinesis and diplotin. So correct answer is C is the correct answer. So leptotene, packetine, actually that is, this is according to this point, but in sequence we are going to say leptotene, what is it, zygotine, packetine, diplotin, diakinesis. But according to these points, the characteristic feature, we are going to give a name, right? So first one is leptotene, second one is packetine, third one is zygotine, fourth one is diakinesis, fifth one is diplotin. Same like that, here we have selected answer. So next, arrange the given statement in the correct sequence. So during prophase 1, now we can arrange it. So what is that? So that is 1 leptotene, after that zygotine, that is 1 zygotine, which is 3, correct, right? So 1 3, and next packetine 2, 1 3 2, and diplotin, that is 4, 1 3 4 5, 1 3, what is it, sorry, 5 4, so 1 3 2 5 4, 1 3 2 5 4. So that is 1 3 2 5 4. So this is a sequence. So this is a 1, you have to keep in mind. So match the column 1 and 2 and select the option in a given code. Synaptonymal complex, we know that always which will be seen in zygotine. So A2, right, and then recombination nodule, packetine, where exactly crossing over takes place because of the presence of recombination enzyme. A2 and B1, A2 B1, and recombination nodule and terminalization of chiasmata. So that is completely takes place in dying of chiasma, I can remember easily. C4, formation of dyad. So what is the meaning of dyad? That is 2 homologous chromosomes with 4 tetrads. So that is, we can say that, so answer for that is telophase. So dyad means 2 cells, so formation of 2 cells, sorry, that is at the end where the deconjugation takes place in the, that is telophase 1. So that is dyad. So dyad cell which has a 2 daughter nuclei, right? So that is what in the telophase 1. So answer for that is, right, A2 B1 C4 and D3. So that is A is the answer. Identify the given figures showing meiotic phase, right? So identify the given figure showing meiotic phases, and here you can say that select correct option. Select correct option. So whenever we are going to say select correct option, that is here the alignment in this A, we can say that they are going to start arranging, that is, and one more thing here you have to keep in mind, these are the homologous chromosomes are there. So that is why we can say it is not belongs to metaphase, it is a metaphase 1 of meiosis 1, that to mainly prophase 1, right? So that is what we can say that the, sorry, not prophase 1, that is metaphase 1. Metaphase 1 metaphase 1 of meiosis 1. So metaphase 1, and here you can say that where the chromosomes start moving, right? So that is chromosomes, not chromatids, that is anaphase 1. And here 2, there's no dyad cell, that is generally. So answer for that is right. So metaphase 1 and then anaphase 1, and here telophase 1. So that is B is the answer. So metaphase 1, anaphase 1, telophase 1. Next, disjunction refers to. What exactly the meaning of disjunction? So here the chromosomes, that is homologous chromosomes, start separate, as we all know that is a characteristic feature of anaphase 1. So separation of homologous chromosomes, that is which forms a feature, what is it, nucleus in the daughter cell. So this separation, so this is arrangement of homologous chromosomes in the equatorial plate. Now they are going to separate. As this separates, so that is where you can say in the anaphase 1. So disjunction, separation of homologous chromosomes. So where exactly separation of homologous chromosomes in anaphase 1 is a correct answer for that. Next. Next question. That is 48 question. Match the column 1 and 2. Select the option from the given codes. So we are going to move towards chromosomes move to equator. Chromosomes move to equator means that is a metaphase. It is not a packaging. Packaging where the crossing over takes place and chiasma appears, right? Egg-shaped structure, right? Chiasma. And this is wrong. Centromere splits and chromatids move apart. Centromere split and chromatids move apart. That is, chromatid is move means that is anaphase, not anaphase 1. So anaphase, centromere splits and chromosomes move towards. Here no centromere splitting, sorry. That is centromere, right? It is not necessary to split, and directly they are going to move with respect to their respective centromere. There are 2 centromere in homologous chromosomes, and these complete chromosome will going to move towards the opposite pole. So that is what the answer. So here this is also wrong, right? So move apart, and C option, pairing between homologous chromosomes. So where you can say that the pairing between homologous chromosomes which takes place, right, which takes place in the zygote. So that is here you have to take. Crossing over between homologous chromosomes, which is packeting. So let us take, what is it, the D1. Here we can write that D1, C2, and chromosomes move towards the equator, that is metaphase, and this is anaphase 1. So answer for that is A4 and then B3, A4, B3, C2 and D1. A4, D3, C2 and D1. So this is the correct answer. Next, during meiosis 1, the chromosome number. So what happens during meiosis 1 generally? The chromosome number which will be there, out of 2 homologous chromosomes, 1 will going to move. So just assume that homologous chromosomes which will be there like this, right? They are in the paired condition, right? So only these 2 will going to move to the one cell, and these 2 out of 4, right? Here you can say that the 2 means A and B chromosome. If you consider that, only this is the A, this is also A, that is homologous chromosome, let me give, and this is a B and this is B. One chromosome of that A homologous chromosome will move, and the other chromosome of B homologous chromosome will move, right? So this is where it will going to receive a, what is it, half, reduced to half. During meiosis 1, chromosome number is reduced to half. Next, during meiosis 1 in humans, one of the daughter cells receives. So what is exactly one of the daughter cell will going to receive during meiosis, one of the daughter cell receives, so where recombination, crossing over occurs, exchange of genetic material. Because of crossing over, what is that? Recombination, and that recombination leads to the formation of recombinants. So what is the meaning of recombinants? Right, a mixture of both maternal and paternal chromosomes will be there. So answer for that is B. So with this we can conclude this chapter, and remaining questions will be given in homework. Thank you.